Good afternoon and welcome to the ICTS Precision Medicine session at Venture Cafe. I'm your host, Rob Culverhouse from the Department of Medicine at WashU. We meet on the third Thursday of the month at 4 p.m. and our sessions last 50 minutes. Our mission is to connect innovative biomedical science generated at WashU, SLU, and MU with entrepreneurs and businesses in the St. Louis region. We hope these sessions stimulate your thinking, and we encourage you to share your ideas and insights with the speakers after their presentations. In case you'd like to follow up with either of today's speakers, you can find contact information along with information about past and future talks on the WashU Precision Medicine website. That's ictspm.wustel.edu. Look under events. Please feel free to unmute yourself when you have questions, and you can also use the chat function. Today's presenters are faculty from the Pediatrics Department at the WashU School of Medicine. Megan Cooper and Jorge de Paolo will tell us about some ways that studying rare genetic disorders can have a broad impact on medicine. By learning the genetics underlying rare disorders related to immune cells and platelets, their work has the potential to target therapies to individual patients, as well as suggesting drug targets that may impact more common diseases. Dr. Cooper's translational research program focuses on understanding the mechanisms of immune cell control, including regulation of killer cell activation and mechanisms driving immune-regulated pediatric diseases. Dr. de Paolo is the chief of the Division of Hematology and Oncology in the Department of Pediatrics, so it's natural that his research focuses on the genetics of bleeding and mechanisms of platelet activation. Some of his discoveries of disease-causing genes have set the stage for potential new therapies for leukemia. So thank you both for being with us today, and Dr. Cooper will start us off. So welcome and take it away, Megan.
Okay, is that sharing going well, Rob? Good. Okay.
Yeah, looks good.
So thank you for that great introduction, and it's my pleasure to be here. This was kind of a fun talk to make because I know that the audience here is very varied, and so it's really, this is not meant to necessarily be a science talk, but just to give you a broad overview of what we're working on in the lab. So I'm a pediatric immunologist, and I treat kids who have rare diseases of their immune system that can cause a number of different problems. And so to just kind of give you a background, I'm going to show you here the story for a few minutes about one of my patients.
My name is Parker, and I will win my fight.
One day, I just happened to be rocking him in a rocking chair, and he was flipped the other way, so he was facing outward. And I was just holding him here, and I kind of felt his stomach, and it just did not feel right to me. So I took him immediately to the pediatrician, and we were there less than 5 minutes. He's like, "You need to go to the ER right now." Nurses were saying, "Oh my gosh, he looks so pale, which I sort of feel guilty about because I guess I didn't notice it. They started running tests. They realized that the spleen was in fact very enlarged. They did the blood count and it was a 4. It's supposed to be about 12. And I guess you see him every day so you don't notice. So I felt really bad.
We knew he was really sick, but didn't really know what was wrong. So that's really a bad place to be in because you really have no idea how to manage expectations.
I am a pediatric rheumatologist and immunologist, and I'm the director of our clinical immunology program. When I met Parker, he was a 3-year-old, and he was a pretty typical 3-year-old boy running around the room. I remember, very rambunctious. He had been treated by the hematology doctors here for about 1.5 years at that time for very low blood cell counts, and he had been getting blood transfusions. In addition to the low blood cell counts, he was having a lot of infections.
How are you?
Good.
You're good? Did you sleep well? Did you cry?
No.
We we were able to identify that he had a low amount of antibodies in his bloodstream. And so we were able to treat that by giving him infusions of antibodies. But that still didn't tell me what was actually wrong with Parker's immune system. It wasn't just that his blood cell counts were low, his immune system was actually destroying his own blood cells. When we got the results back, it came back positive for a mutation in a gene called PI3 kinase. And interestingly, if we had sent this test 6 months earlier, we would not have identified this as the cause of his disease, because this was a brand new disease that was just reported at the time that we sent his genetic testing.
When they started talking to us about the diagnosis, there was so little known about it. It was so rare that there was less than 60 people in the whole world that had it. So while they know what it is, they don't know what to do about it.
And Parker's body just can't make antibodies appropriately. So we give him an infusion of antibodies. He gets this once a week at home. This is something that he is going to require for the rest of his life to help to keep him healthy. Our donor-funded research here at Children's and Washington University is essential for exactly this type of work, disease discovery in young children.
It was a mission to her to figure it out, and she did, and I'm so grateful to her. I know that's her job and that's what she wants to do, and that's probably her passion, but it's my kid, so to me it means everything.
And there is no other who loves me the way as the you do dear.
My dear mother.
So that is Parker's story, and I thought that nobody could tell it better than Parker and his parents. And I just wanted to give you an idea of what types of patients we are treating and we are seeing, because these are very rare diseases and something that really most people are not familiar with. This is, what Parker has, and what I'm interested in my clinical practice and in my laboratory are what we refer to as inborn errors of immunity. So these are genetic diseases of the immune system. We also call them primary immune deficiencies as well. And there's actually, so Parker has one specific type. There's actually more than 475 different diseases known at this point. And I'll talk a little bit about that genetic discovery. They typically present in childhood and rare, but collectively affect about 1 in 1,000 kids. So while Parker was one of only about 60 kids in the world diagnosed with that disease, when you look at all the 475 diseases, inborn errors of immunity are actually a pretty significant problem of childhood. And the other thing that I really wanted to just impart today is that these kids, these families, they have a lot to teach us about the human immune response by investigating the exact and precise molecular mechanisms of their disease. And on the right here is just a little Venn diagram kind of showing what some of the different symptoms of inborn errors of immunity are. And my lab is really interested in these syndromes that kind of combine multiple different clinical phenotypes. So these patients tend to have defects of their immune system that cause infection, as you saw in that little video. They also have autoimmunity. So in that case, that patient, as we described, his immune system was actually destroying his own red blood cells. And then they have a pretty significant risk of lymphoproliferation and cancer due to these immune system defects. Over the past couple of decades, there's really been a surge in identification of genetic causes of inborn errors of immunity. And this, says that this is from 2021. This was actually written in 2020, just got published. And this is already out of date because we're already kind of like up here probably by now. But this is just to show you the total number of monogenic immune deficiencies. And when I say monogenic, I mean we can identify one problem in one gene that is causing the problem with that child's immune system. And so you can see that there's been a really rapid rise, and much of this rapid rise, particularly over the past decade, has been with next generation sequencing techniques. So meaning that we now have the ability to rapidly and accurately sequence all of the coding genes that we have. So there's multiple implications for human health. So, for the patient, and this is really what my interests are, primarily because I see kids like Parker in clinic every time I'm in clinic, and I'm thinking about them and their families and how I can provide them a better diagnosis and treatment. And so if we can have a molecular diagnosis, that can actually provide a target for treatment. So the particular disease that we opened up with, which is a disease of a complex of molecules that we call collectively PI3 kinase, but the specific gene that you saw in that video, PIK3CD, is what is the problem in this disorder. So what we know is that this genetic change leads to a gain of function or extra function in the protein encoded by this gene. And that protein is called P110 delta. So this extra function leads to the immune cells not being able to control their responses and leads to that susceptibility to infection, autoimmune disease, and cancer. And there is actually an ongoing clinical trial of P110 delta inhibitors in patients like Parker. So this is a clinical trial that's going on at the NIH. It's, I think that particular trial is actually the only trial, and it's only for kids age 12 and older because of some age restrictions. But I want to point out that this drug is also in clinical trials for other things like lymphoma and also autoimmune diseases. But, from the bigger picture standpoint, as a physician and as an immunologist, I think that discoveries from these patients really also have the potential to give us insight into more common disease. So I'd like to point out that there's another disease called X-linked agammaglobulinemia, and the gene for that disease is called BTK, and that gene was discovered in 1993 as the cause. And so this disorder is something that affects boys, and these are boys born without a part of their immune system called B cells, and B cells are antibody-producing cells. So these boys are born without the ability to produce any antibodies, and I think we're all very aware of the importance of antibodies in controlling infection with everything that we're hearing about COVID now. So this disease and this discovery that BTK was behind the disease actually led to the use of BTK inhibitors as standard of care therapy for chronic lymphocytic leukemia, which is the most common chronic leukemia of adulthood. And there's over 20,000 new cases each year. So they discovered this gene in a very rare subset of patients born without B cells. So they discovered, hey, you need this gene, you need this kinase to actually promote B cell differentiation and survival. We could actually use this as a target to treat a totally different disorder where there is a cancer of B cells. And so that's what's done in CLL here. So again, just highlighting that these genomic discoveries advance our understanding and can provide targeted therapies for both our patients, so the rare disease, and then also potentially for larger groups of patients with more common disease. And what we do in the laboratory is we try to identify genetic causes of inborn errors of immunity. And for us, it all starts with the patient. It's typically patients that we see right here in St. Louis. Sometimes it's patients referred from other institutions that come here, or maybe I just talk to their physicians or the patient, and then they send samples. And our first step is genetic investigation to try to discover a disease-related gene. So we all have about 25,000 genes, and what we're trying to pinpoint is that one point in one gene that might be causing that patient's disease. This can be a very long process. It can take months to years. After you have a candidate gene, then we work to actually validate that gene. So we want to say, okay, there's a change in the gene, but does it actually alter how the protein encoded by that gene works? Is there actually a functional consequence to that? And then once we validate that, we want to say, okay, it's not working well, but is that really what's causing the problems with the patients? And usually to do that, we put together some cohorts of patients, and I'll talk a little bit about that at the end. And then what we want to do after we can identify the precise cause of a problem is we want to take that information back to the patient and think about how can we actually change their therapy and provide precision therapy. So to give you an example of that, this is a disease that we discovered in 2015. It's called Stat3 gain of function. So this is another example where a genetic change leads to increased function of the protein. In this case, this is a signaling protein of the immune system called Stat3. This was our very first patient here in St. Louis, and he has arthritis. You can see that in some contractions of his hands, how he's holding his Lego board here. He has contractions of his elbows. He's on a very high amount of what we call steroid medications, and that leads to this enlargement of his cheeks here as a side effect. So over here on the right, this is years on the x-axis, so 0 to 8. So this is his diagnosis until year 8. This is his hemoglobin. This is a marker of red blood cells, and normal should be about where this dotted line is here, somewhere around 12. And what you can see is his hemoglobin was very low. In fact, it sometimes dropped dangerously low. So down in these regions here, that's him being in the intensive care unit, very dangerously low. And we were able to bring it up, but up here, what we were using were medications, steroids again, and then also a medication called rituximab here in green that would help him. But really, these are very non-specific medications for this therapy, and they were only temporary. So once we diagnosed this patient with Stat3 gain of function, we were able to provide a more targeted therapy that was already on the market and used for other diseases, and that's a drug called tocilizumab. And this inhibits the upstream activation of Stat3. And so what you can see in red here is once we got him started on this medication, we're able to stop these corticosteroids for the first time in 6 years. He did not require any further doses of the rituximab, and he had stabilization of his red blood cells. So this is really an example of where we get a sample from a patient, we identify a gene, we functionally validate it. And in this case, when we reported this disease, we identified 12 other patients around the world to kind of describe this as a new disease in a cohort. And then we're able to take that information back to the patient. And then this is just to give you an idea of our most recent discovery that just came out a couple of months ago. This is a disease that we call infiltrate. And this is caused by a genetic defect in a gene encoding a protein called TLR8. This is another signaling molecule of the immune system. This is a signaling molecule that should kind of sense danger, but what we see in these patients is that it's always turned on. And so it's leading to this very inflamed state in the patients. And they have a number of different clinical problems. They have low levels of blood neutrophils, which are important immune system, important part of our immune system to fight infection. They have defects in B cells, again, those antibody producing cells. They have a lot of activation of their T cells in what we call lymphoproliferation. This is a CT scan, and this is a very large spleen right here. It should be about this big, and you can see it's down to the pelvis here. And they need bone marrow transplant to treat this disease. So this was another example of a patient that we had here that we performed genetic testing on, and were able to identify a cohort of 6 children in this case. Now here, we don't actually have a targeted therapy that we can use for these patients because there's nothing on the market that alters TLR8 signaling. So this is my last slide. I know that the spirit of Venture Cafe and the audience here is to kind of think about partnerships between potentially academics and industry. And, really, we're not, actively working on any translational studies such as that now in terms of treatment, but I actually think there really is the potential to engage industry partners potentially. So I think that a couple of things. Our patients, a lot of times they don't have a therapy that can be used. So for Stat3, we were really lucky we could repurpose a therapy. For our TLR8 patients, we don't have a therapy we can repurpose. So I think that there's the potential to develop therapies that are highly targeted to the precise molecular defects in our patients. And so there's really the potential to improve the lives of the patients. There is also the potential to translate this to other conditions. So as I showed you, the PI3 kinase, using that inhibitor is we think, has potential to be a benefit for that very rare disease, but it also has potential to be a benefit for more common diseases like lymphoma, where it's being clinically tested. And as an example of how this might work, for example, there's a drug called canakinumab. It inhibits this protein of the immune system called IL-1. And it was originally approved for this very rare disease called cryopyrin-associated periodic syndrome. This is a very rare inborn error of immunity where kids get fevers and rashes and arthritis. And a drug was approved for this because it was very specific to the mechanism of that disease. And it's a recombinant antibody that's given as an infusion or injection. However, after FDA approval for this indication, it's now been approved actually for 6 other similar diseases, including usage in, I don't think it's quite approved yet, but it has potential usage for diseases like gout, which is obviously a much more common disorder. So I think there has been interest in industry in developing drugs for these very rare patients where you could potentially get approval for the drug for a very rare indication, and then you could extend that to other more common conditions where there might be similar pathways at play. So that's all I have, and I'm happy to take any questions now, or if we want to chat after Jorge talks, whatever people would like.
Well, thanks, Megan. So we can give people a couple minutes, just see if they have any questions. But one question I had was just, you mentioned that the last disease you said there wasn't any medication that would directly address that. Is it, are you finding these large medication databases that are now out there in the world? Do you just regularly mine those to see what they do and what off-target effects they have to see if there's something that might work?
Yeah, so we have actually in the lab tried some inhibitors of TLR8, and actually the inhibitors that are available don't actually inhibit our patient's TLR8. So suggesting that our patient's variants are working by a mechanism that bypasses that inhibitor. So I think we would probably need something a little bit more specific to the mechanism whereby there's increased function in our patients. It's also, you know, I treat kids, and so it's a challenge thinking about, you would much rather use an FDA approved drug, right? In terms of like getting up a new clinical trial. But certainly I think that there really is potential to develop new therapies for these patients.
And I see there is a question in the chat. Esma asks, do they also have a defect in FAS and the FAS-L pathway?
Yeah, so this is like a cell death pathway. So our TLR8 patients do not. The Stat3 gain of function patients, some of them do have defects in FAS-mediated apoptosis, which is a form of cell death.
Dr. Cooper, I have a quick question too. Thank you for your presentation. So I wanted to go back to Parker's story, because you said briefly in the movie that the timing of a few months when you did that sequencing was kind of, can you say a little bit more about that? Like, was there a sort of accumulation of evidence up to that point? Would you, if the timing had been different, would you have been more part of the discovery? Just tell us a little more, because that was interesting to hear.
Sure, yeah, no, it's really interesting because so his gene actually got discovered through clinical exome sequencing, which we're very commonly doing now. And so what happened was the challenges with clinical exome sequencing is they are only going to look at genes that are known to cause disease. So about 6 months prior to us actually sending that clinical test, his disease had never been reported. And so it never would have come up in the clinical sequencing. But I think even for research sequencing, it would have been challenging, because there's a real challenge when you only have what we call like an N of 1. So one patient with a genetic variant, and how do you know that that variant is actually causing disease? And so there comes real power in putting multiple patients together, and that actually takes a lot of collaboration. So for like our Stat3 story, we had 13 patients. It was 7 institutions between the US and Europe to gather those 13 patients that were there. And then that obviously didn't happen overnight. And so that's that really kind of just maybe emphasizes the need for collaboration in this field. And I'm sure Jorge experiences the same thing.
Thank you.
So if we don't have any other questions right at the moment, people who haven't had a chance to ask their question yet might circle around after Jorge's talk. So Jorge, welcome, and take it away.
Thank you, Rob. I appreciate it. Thanks everyone. Thank you for coming to my talk, and I really appreciate the invitation. I think that this is such a fun forum, and Megan did a fantastic talk, so it's going to be hard to top that one, particularly the video. I don't have videos. But what I'm going to tell you the next 15 minutes is a couple of things. I'm going to give you a background on why coagulation and platelets are important, and you're going to realize that they're very important, actually. I'm going to give you a couple of vignettes on how, I was thinking about this, you know, on what you guys are trying to do here, and how discovery of rare coagulation and platelet disorders led to the development of drugs, for example. And then I'm going to tell you in the last part what we've done what we are doing in our laboratory, trying to emulate those kind of discoveries. And as Megan post it at the end. I'm gonna post some of the questions that I have or the opportunities that I think that there are for this sort of interaction between academia and industry. So platelets are good. They can be good. And I can show you here, how there are a bunch of red cells that are coming out. Let me see if I can get my pointer. There are a bunch of red cells that are coming out of a blood vessel that has been punctured or some sort of injury. And then platelets, what they do is they help those red cells and other things to make a hemostatic plug. So that's when platelets are good for us. But platelets can be bad too. And what happens, when sometimes you have a similar injury, maybe not as dramatic, and then platelets help you to make a thrombus. And if that thrombus or that clot occludes the whole vessel, that is not good for you. So that's what I call the bad. And eventually they are doing their job, but this is a very delicate balance between these components, and therefore they can be bad for you. And they can be ugly too. I remind everyone that cardiovascular disease is the most common cause of death in the world still. And platelets actually, which are forming here, white thrombus on top of atherosclerotic plaque, are gonna be the last event that happens in people that have myocardial infarction, and people can die of this disease. So again, platelets belong to this axis, and coagulation factors too, which is what I call the good, the bad, and the ugly. So how do if we dig a little deeper in how they work, I tell you, this is a blood vessel wall. These are the platelets. This is von Willebrand's factor, which is like the mortar that allows the platelets to stick to each other. And if you have an injury in the blood vessel, as is shown here, you're gonna have those platelets to come and von Willebrand's factor to come and make a plug. But then you're gonna trigger the coagulation cascade, which has innumerable components that you don't have to remember, but eventually the whole idea of the coagulation cascade, or the whole goal of the coagulation cascade, is to form this molecule that is called thrombin, which is the most potent procoagulant molecule that you're gonna have in your body, and eventually make a fibrin clot. Because our bodies are smart about this, our systems are smart. We have an anti-thrombotic system that clicks in right away in order to avoid that occlusive clot. And at the same time, we have a system that's gonna come and re-architecture, they're gonna change the architecture of the clot. So as you can imagine, bleeding and clotting is a fairly delicate balance that eventually, if it's overdone, you can get into trouble. So I wanted to point out to 3 investigators actually from Washington University in St. Louis that really is studying rare diseases. They're all disease. They are kind of my hematology heroes here. Really this happened in our campus, made us understand how this whole system worked. One is Phil Majerus, who discovered how platelets work, and that giving low dose aspirin to patients, discovered how aspirin works in platelets, and giving low dose aspirin to patients will prevent thrombosis and myocardial infarction. Dr. George Brose, who discovered 2 different molecules in this system based on studying patients with rare diseases. And Dr. Evan Sadler, who discovered how this molecule, particularly von Willebrand factor, is cleaved, and eventually discovered the underpinnings, or the molecular underpinnings of the disease that we know as TTP. So this has been going on in your institution, which I'm new to, for a long time. So where are platelets made? Platelets are made in the bone marrow, and they're made from these gigantic cells, probably one of the largest cells in the body, that are called megakaryocytes, and they are the platelet factory. And they come from these, what we call hematopoietic stem cells. They then mature because of a cytokine called thrombopoietin into megakaryocytes, and then they go into these proplatelet forming megakaryocytes, where you see these extensions that eventually is gonna pinch off platelets into the circulation, okay? So that has to happen in a fairly coordinated way. We make approximately 1,000 billion platelets a day, that to maintain an approximate number of 150,000 to 450,000 platelets per microliter of blood. So it's a fairly coordinated mechanism. So how does the platelet work? The platelet has numerous receptors, and if you stimulate the platelet, you're gonna have calcium mobilization, and you're gonna activate this molecule that I'm putting here, the alpha-2 beta-3 or fibrinogen receptor, which is what allows platelets to aggregate with each other. And then that triggers more activation. You spread your platelet, and then you release the granules. And that's what the platelet do eventually, trying to act in an injury in the blood vessel. So that particular receptor, alpha-2b beta-3, which is what is called an integrin, when that receptor is gone because of a genetic defect, you have something that is called Glanzmann thrombasthenia. And that's my first vignette about how this discovery of rare diseases can take you into new frontiers in terms of therapies. So this is a classic example from how things went from the bedside to the bench and they went back again to the bedside. So in 1918, Edward Glanzmann, who was a pediatrician actually, described what we call thrombasthenia, which means weak platelet. There he is. So it took another like 60 years for Alan Nerden and Jacques Kahn in France to notice that the absence of that receptor that I shown you was the basis, the molecular basis for Glanzmann thrombasthenia. Then in the early 80s, those receptors were cloned and the genes that encode for these receptors were discovered and cloned. And then a very smart investigator at Rockefeller University in the 80s and 90s started to try different monoclonal antibodies against this receptor because it was discovered through that. That was Barry Kohler at Rockefeller. And he patented this antibody, and RioPro, which is called abcipsimab, was based on this particular discovery, and approximately more than 10 million people with an unstable angina, which is a cardiovascular problem that can lead to death, have been treated in emergency departments in the United States and the world with this particular drug. I think that's a classic example of how these rare diseases can take you into new therapeutics. And again, the second vignette is something that I thought that historically is very interesting. As you can see here, this is the pedigree of famous Queen Victoria of England. And as you can see, for us geneticists, we love to draw pedigrees. There are here people that are in solid symbols, and that usually means that are affected by the disease. As you can see here, these are hemophilic males. This is hemophilia. And then the females that have half a symbol or half a circle, a solid circle, are the carriers. So as you can see there, Queen Victoria, gave rise to eventually the most famous hemophiliac, probably described as was Alexander, the son of the Tsar of Russia. And eventually, interestingly, this probably changed the history of the world because the influence that Rasputin had in this family, people think that is because he was the doctor that treated hemophilia approximately in the late 1800s and early 1900s. So since then, as you can see here in the 60s, people were treated before with, blood transfusions. But then researchers became smarter and they identified the factor that was missing. So they did these plasma concentrates. And then they were giving patients plasma concentrates. But then, as you can see here, all these plasma concentrates were infected with hepatitis B, hepatitis C, or HIV. But by the mid-80s in the United States, 80% of the hemophilia patients were HIV positive. So that was a tragedy that eventually propelled research. And the factor 8 and the factor 9 genes that cause the deficiency cause hemophilia were cloned then. So therefore, companies came in and they started to produce these in the lab. And as you can see here, different cell cultures produce factor 8 in industrial proportions. You can see here a bioreactor factor 8 production, and that's how patients started to be treated with recombinant factors. And we haven't had in the United States any case of viral related transmission in patients with hemophilia since the late 1890s. So how do we take this kind of historic vignettes to our laboratory research over the last 20 years? Well, we try to do similar things. And as Megan was telling you, we do something similar. We make sure that all our patients have an adequate phenotype, which means to really determine the clear diagnosis of that patient, even if we don't know the cause of the diagnosis. In her case was the deficiency of antibodies. In our case, it's bleeding, for example. Then we phenotype their cells. We want to know if their megakaryocytes are normal or not. And then we try to measure those platelets functionally. And eventually, we go back to these families that we have and these rare patients, and we do this high throughput sequencing, and which is next generation sequencing, trying to find mutations. And as you can see down here, this is a continuum, because once we find a variant, we really have to prove that that variant is causing this particular phenotype. So then we go into what we know well in the lab, which is study cell models, study animal models, and doing genome editing of cells and see if we can replicate the phenotype that we saw. So I remind you how platelets are made again, but the next two cases that I'm going to give you, and there are two slides, are related to families that we studied in the past. You know, the first one is a family that we studied. It's a Native American family that we studied in the state of Iowa when I was at the University of Iowa. And as you see here, as I told you, we love to draw pedigrees. And this is the affected individuals, the one with the solid symbols. And what these patients had is something that had been identified before, which is called gray platelet syndrome. And what that means is, like, if you see here, this platelet in this patient is very different from the platelet from her father, which is like much darker. And if you take that into the electron microscopy realm, you can see that these dark granules that these platelets have are not present in the girl. And that's what is called gray platelet syndrome. So that was in the year 2010. We did several experiments, and it was really hard to sequence, but eventually we sequenced platelet RNA, and we found that mutations in a particular gene that hadn't been described at that point, just identified, were responsible for these. And then we did what, we sequenced a bunch of other families, and we did what we do well in the lab, and we created an animal model. And the animal model has the same sort of platelets that this girl had. So therefore, now we have a model that, and I'm not going to show you all the publications and experiments that we've done, that is really allowing us to understand what is the role of these granules in platelets, and see if, because these granules are the ones that are very pro-thrombotic, that activate the platelets, and see if we can design therapeutics or things that can block those granules for not forming, but for releasing their content and eventually as antithrombotics. But at the same time, these platelets have very low platelet counts, trying to understand how this gene is involved in the production of platelets. The second family that we had was a family from Detroit, actually, that we flew from Denver, where I was at that point, to recruit this family in our studies, where, as you can see here, they have solid symbols, as I shown before, are the ones affected. They have low platelet counts. These are the unaffected, that this is the affected. And then we found two more families that were similar to this, one in Czechoslovakia and one in Italy. As Megan was saying, this is usually the result of the collaboration that is international, because you need a lot of these families. And these families have something interesting. Not only they have low platelets, but as you can see here, their asterisks, those patients develop leukemia. So we thought that we were in the presence of a cancer predisposition syndrome at the same time that they have low platelets. When we did bone marrows on these patients, this is the normal megakaryocyte. That's the platelet, the cell-forming platelet. And as you can see here, this is a very different megakaryocyte, much smaller, indicating that these families, probably the patients that were affected, had an abnormal differentiation of these big, the hematopoietic stem cell into megakaryocytes. And we found the gene, among other groups, I had to give credit to, in a transcription factor, which is called ETV6. And again, this underlined the fact that when, for example, when people in St. Jude Cancer Research Center went and studied all their families they have with isolated leukemias, and they went to their germline, which is the DNA that you were born with, they found that 1% to 2% of patients had mutations in this gene. So eventually, I think that our discovery underscored an important cancer predisposition syndrome, but also for what we like to study is why are these genes affecting platelet production? And so as I shown you before, if you have from hematopoietic stem cell, you give this cytokine that is called thrombopoietin, you have a late megakaryocyte and this proplatelet forming megakaryocyte. We can do this in the lab now. So these are CD34 cells that then we put on thrombopoietin. We can make these late megakaryocytes, and then we can make these beautiful proplatelet megakaryocytes that eventually are going to form platelets. And now we are getting to the point that we can study those platelets, so that they make enough that we can study those. Why? Because we have this new technology called CRISPR-Cas9 that you might have heard of, and we can genomic edit in these cells and eventually insert the mutations that we found or put other mutations and see how they affect this platelet production. So as a last slide for you, and kind of mirroring with what Dr. Cooper did, because I saw her last slide and I say, okay, I'm gonna do the same. What are the potential to engage industry partners in our research? Well, I think that if you understand the molecular mechanisms and genes involved in platelet production, you can increase your ability to make platelets in vitro. And there's already one company that does that, founded by a good friend of mine, Joe Italiano in Harvard, which is called Platelet Bio. And I think that they founded this company 8 or 9 years ago, actually. And they're trying to get that holy grail of making enough platelets in vitro, that eventually, if you have a patient, for example, in the future that comes for a surgery, I know that it's going to be a bleeding surgery, and the patient is going to need platelets, you might bring this patient a couple of months before, you extract some cells from the patient and eventually manipulate these cells in these bioreactors in order to make platelets, and the patient will have a self-transfusion with the platelets of his own body. Again, this is a little science fiction now, but I think that that's going to happen in the future. And then eventually the other thing is that because we studied all these platelet pathways, if we understand how these pathways that are affecting patients, and I didn't show you many of the patients that we have with rare diseases in platelets, will really improve our ability to generate antiplatelet agents. So excuse the typo there, but the reality is that still patients are being treated with aspirin, for example, and maybe Plavix and other new drugs, but we are lacking a lot of new drugs that make the patients not clot and avoid the bleeding.
Okay, well, I hope to see everyone in April when we'll hear from Dr. Justin Sachs, who's chief of the Division of Plastic and Reconstructive Surgery, and Dr. Matthew McEwen from the Department of Neurosurgery, both at WashU. I want to thank both of our speakers again.
Thank you.
And I hope you get some questions.
All right.
Anytime.
Thank you very much.
Yeah.